Please welcome Alexandra Huener, Milner Therapeutics Institute.
Hello everyone, I'm Alexandra, the head of entrepreneurship at the Milner Therapeutic Institute, University of Cambridge. We are a research institute and a global outreach program. Our key strength is combining academia and industry to accelerate pioneering science into therapies. Within the Milner, I'm leading the incubator Frameshift at Europe's largest biomedical campus, and we nominated Enhanc Genomics for the following reasons. Enhanc has developed a platform to profile 3D genomes. This will be the game-changing platform in the era of precision medicine and will have a profound impact on human health. Enhanc is fast-growing. It has established key partnerships and also makes rapid development process. It's also a driven company with a great culture cultivated by its CEO. Please welcome with me Debora on stage.
Breaking the wall to 3D genomics for therapeutic development. Debora Lucarelli, Enhanced Genomics.
Thank you, Alexandra. So, DNA is the code of life. Mark these words. Carolina and Marina have already done my pitch. We all know that drug discovery is expensive, it takes a long time, and there is a high percentage of failure. But those who really are failed are the patients. And that's because drugs tend to be of very wide actions and not on targets. Why is that? The reason for that is that we all are genetically very different. And Enhanced Genomics is bringing a new viewpoint, the 3D genomics perspective, that allows us now to look at the entire genome in its entirety and bring a solution to some of these problems. Here is an image that everybody's familiar with. We have our double helix made of 3 billion of base pairs. How many people in this room know that we utilize only 2% of these informations? The protein coding. So much for precision medicine, so much for personalized medicine. The remaining 98% constitute here areas like this called non-coding DNA. And this is where the overwhelming majority of all our mutations sits. Every single mutation sits in this space. We can't develop therapies, we can't action them. There isn't much to do because we don't know the function of them. So this is actually not even a real image of our DNA. So if we really think about it, this is what our DNA looks like in the cells. So it's tightly folded, highly compacted, and it's very complicated. I'm sure you agree with me that this is very different. And it varies from cell to cell, from person to person, and from disease to disease. So to simplify the challenge and also the solution, we look at the folding on every single loop where the mutation come into contact with the gene that they control. And we do this for every single cell type across the entire genome, and we can do this for every disease. Biology is complex, and there is no surprise that we need better tools. So Enhanced Genomics has developed a very elegant platform that is a combination of a proprietary molecular assay as well as a number of algorithms. And what we use it for is to collect, measure actually, sample from humans, diseased and healthy. We identify very complex biologies, and then we have a pipeline of targets, biomarker or targets, and we validate them within primary tissues. We have validated this approach. We have a variety of partners, and one of them has actually nominated us here, and we have just finished our first commercial partnership. How we differentiate from other people in the field, other players? We made these decisions that we will be actually profiling and deeply well-characterizing only primary tissues and create the resource in-house. And we look at targets that are specifically identified by 3D genomics interactions, that they are really long distance and can be identified only with this platform. Other omics data can't do that, and therefore we have a first look at some of these pathways. So the method is well established, and the team has pioneered the field for more than 2 decades. We have more than a lot of publication in very high-profile journal. We are licensing 2 patents, and the company has just actually filed for 2 more. We have demonstrated diversified funding, and we actually started our commercial traction. So we are expecting to go into fundraising starting next year. And what is the purpose of our fundraise? Where are we going to be? What we want to be is actually to further the clinical development of our 2 internal programs, and we want to expand the infrastructure and take some of the leads a little bit further ahead. We want to continue the development of the platform, but we also want to strengthen and expand the number of partnerships that we currently have, and we want to establish some new ones. I'm joined by a deep tech team of plenty expertise, as well as many visionary and well-established entrepreneurs. And together, we are paving the era of 3D genomics for real precision medicine. Thank you.
Great. No, don't come too far. Come here. Yeah. We want to see you. We want to see you on camera. I see already 1 question here. I see 1 question here. And I see 1 in each block nearly. OK, let's start with this block, please.
Thank you. How do you actually establish, just a few words maybe, how you establish the 3D structure? And actually what you're saying is that there is a particular 3D structure specific for a particular disease? Is that true? Is that what you're saying?
Yeah. So the conformation of our DNA, the long linears that you see, actually, this is your DNA, actually. In reality, in our cells, is very folded and tightly compact. So what we do, actually, is we have a technology that allows us to chop these massive blocks in many small pieces, and then we actually detect all the interactions between the genes and the promoter that control them. So that's the first approach that we are doing. We then obviously using sequencing methods that allows us to look at the nature of these interactions. We do this genome-wide so that we can actually have pathways and not just single targets. The reason this is different from disease to disease is that this folding will change from disease to disease and from person to person. Therefore, we need to look at a multitude of parameters in order to find the right path.
OK, we have 3 questions waiting.
Sorry.
So we'll take Chaz first, Frank then, or Frank and Chaz, as you like. You can decide.
Thank you. I mean, the question is, do you have a therapeutic focus or a disease focus?
Yeah.
I was just conscious
Yes. Yeah,
nucleome.
it's not generic. No, it's not. Sorry, you're conscious of...
I was going to say nucleome do
therapeutics.
something similar.
Yeah.
And
Yes,
so I don't know what the difference is.
So first of all, the difference is that there are 2 or 3 platforms around, and we all use a slightly different business model. And the technology is also slightly different. So we obviously profile the entire genome, while they can't. So our focus started initially because we started a collaboration with Janssen, and therefore we started with lupus in the first instance, and we applied the platform for patient stratification, because possibly this is the most important step at the beginning in order to identify the right therapeutic approach. After that, we have now developed more in the MS area, and we have a project as well through, obviously, again, collaboration through the tumor microenvironment. So those are our first 3 large programs.
OK, Frank, please.
The first question you already answered regarding patient stratification. That's something I fully understand.
It's the most important
because
thing.
that's already ongoing. Genomic profiling of patient population is more or less standard also in clinical trial. But the second part, really to identify a new target in the non-coding DNA. Do we have already an acute
Yes, we do have a variety.
Example?
Yes, we do. We do have already identified about, we have a pipeline of these targets. What we do, we modulate them in the very first instance, some of these non-coding regions to demonstrate in primary tissue that these are going to have an effect. We measure the effect of these particular targets over certain genes and over a specific area.
My question is, do you already have an example that with a small molecule, you can specifically target the 3D structure of the DNA? Because targeting the 3D structure of RNA, something like that is ongoing, but it's very, very difficult.
So there are
24 seconds.
2 or 3 approaches. So one of them is the gene editing approach, and the other one is to use antisense oligos, or the last one we are trying at the moment is the AAV vectors.
Okay. I'm sorry, I think we have 10 seconds. I'm very sorry, we cannot take the
I'll come and find you afterwards.
Yes, that's a very good idea.
So, yes, thank you.
This is Angelica. Please, you can go. Thank you so